The overall goal of this procedure is to monitor cells and tissue through a non-contact and marker-free method. This is achieved through Raman spectroscopy, a laser-based technology that detects the inelastic scattering of monochromatic light, producing a typical spectral pattern for each biological sample based on its inherent biochemical composition. Using a standard fluorescence microscope coupled to a Raman spectrometer, direct comparison of bright field and fluorescence images with Raman spectra is used to monitor numerous biological samples. Each chemical vibration is assigned to a specific Raman band, and the peak intensity is correlated with the amount of the present molecular bonds. Similarities and differences of the spectral datasets are detected by employing a multivariate analysis. The resulting spectra can be used to check the integrity of tissues after harvesting a biopsy, or to prove the sterility of isolated cells. In addition, this technique enables the characterization and identification of different cell types in vitro. Ultimately, Raman spectroscopy is a promising laser-based technique for tissue engineering and clinical applications through non-contact, label-free analysis.
Demonstrating the procedure will be Daniel Carvajal Berrio, a technician, and Miriam Votteler, a PhD student from my laboratory.
The main advantage of this technique over existing routine methods, such as histology and immunofluorescence imaging, is that no sample processing is required.
Though this method can provide insight into single-cell state and extracellular matrix components, it can also be applied to multicellular tissues and organs.
To prepare customized Raman